Dear friends, welcome back to my channel Easy Quality Learning with another video, that is media fill study. So this is also called as aseptic process validation or media fill simulation. So different names are there. So let us see what is there in this particular presentation. So contents of this presentation will be guidance documents, significance of media fill, prerequisites of media fill, elements of media fill, and execution of media fill. So first we will see what are the guidance documents which are available for this particular activity. So first guidance is US FDA CDAR guidance, sterile drug products produced by aseptic processing. So this is US FDA guidance, and in this the different parts of aseptic drug processing are given, in which aseptic media fill is also mentioned in this particular guideline. Then second one is WHO TRS 961 Annexure 6, good manufacturing practices for sterile pharmaceutical products. So this is WHO guidelines. Then PICS is having one guideline, that is PE 009 Annexure 1, manufacture of sterile medicinal products. Then HPFBI Canada is having specific guidelines for aseptic media fill, that is process validation, aseptic process for pharmaceuticals. So this is a guideline of Canada. Then EU GMP Annexure 1 is available, where manufacture of sterile medicinal products is there. This is a European guideline in which how to process the sterile products it is mentioned, and one part is for media fill or aseptic processing. Then comes significance of media fill. So why we need to do the media fill? What is the significance of it? So first, this is applicable for your sterile products only. This is not done for non-sterile products. So whatever sterile preparations are there, like small volume injection, dry injections, large volume injections. So for this only this media fill is applicable. It is not applicable for non-sterile products, and this is used to evaluate our capability to do aseptic processing. So our design, our qualifications, they are all capable of doing the aseptic processing or not. It is just a parameter to evaluate all those things. In other words, you can say that this is a process validation for aseptic processing. Then demonstration of aseptic processing in critical area is critical area for sterile dosage form. Since as you know that in sterile preparations, all the preparations they go directly into the bloodstream. So if there is any sterility failure or any ADR is there with reference to sterility part, then it may create a great problem to patients. So that's why it is very important with reference to patient health also. Then next is prerequisites of media fill. So if you want to do a media fill, then what are the things you need to have in advance with you, or what are the things which need to be prepared before doing media fill? So media fill protocol shall be signed off. So whatever activities we are going to do, as per that we need to prepare a protocol, and that protocol should be signed off before execution of or before start of your media fill activities. Your HVAC, heating, ventilation and air conditioning system, or EHUs, which are there for your sterile processing and accessories area for the sterile processing, they should be qualified. IQ, OQ and PQ for all the AHUs and HVAC system shall be completed. All the UDAF of a sterile area shall be qualified. Means whatever UDAF we are using for your filling point or filtration point, or your material unloading point, or in your washing and material preparations, we might be using UDF for cleaned containers or cleaned vials. So for that also the UDF shall be qualified. Then your all the critical equipments which are used in aseptic processing, like your steam sterilizer, deparination tunnel, then your filling machine, bunk processing machines. So these all machines shall be qualified as per their requirements. All the operators shall be qualified. All the operators who are going to participate into your media fill, they should be very well trained and qualified, and they should be explained how the activities has to be done during aseptic processing. Then walk-in incubators or incubators, whatever we are going to use for incubation of media fill units, that should be pre-qualified before your incubation or start of media fill activity. The media which we are going to use, that should be qualified before start of media fill. So these are all the prerequisites of media fill which should be fulfilled before start of media fill. So let us see what is next. So next is elements of media fill. Elements means what are the things we need to consider during media fill. So selection of growth media, selection of longest run, selection of container closure system, line speed, simulation of all the activities in aseptic area, inclusion of all interventions, participation of all operators, then units, number of units to be filled, incubation temperature and duration, and interpretation of the results. So all are these subparts of the media fill activity which need to be taken care when we are making a protocol and whenever we are deciding for the media fill activity. So let us understand these things one by one. So first is selection of media. So as per industry trend, soybean casein digest media is used for media fill activity. Then what are the prerequisites of media? Media shall pass through in the growth promotion test. It should be demonstrated that whatever media we are using, it is passing in growth promotion test and it is capable of growing the low level microorganisms. In media qualification, all the categories of organisms shall be selected for growth promotion test, which includes your aerobic bacteria, yeast, fungus and environmental isolates. Generally 10 to 100 CFU suspension shall be used for growth promotion test, and within a stipulated time we should be able to get the growth of the media. Longest run and container closure. So media fill duration shall be selected based on the routine production shift. So if our production is running in 1 shift or 2 shifts or 3 shifts, based on that, whatever is the longest run for that particular container closure system, we need to consider that longest run for the media fill, so that your shortest duration as well as longest duration shall be can be accommodated in media fill activity. And container closure, which is used for our commercial activities or production activities, same container closure shall be considered for your media fill activity. Then longest run shall be selected, which we have already discussed. Then line speed and environmental conditions. So high line speed with frequent intervention shall be used. Slow speed allows the prolonged exposure of the media. So in both the conditions, some advantages and disadvantages are there. So we can use the combination of both. Sometime some part of the media fill we can use high speed and some part of the units can be filled at low speed, so that we should be able to cover the range of line, so that we should be able to cover the speed range of our production line. Then same environmental condition shall be maintained as it is maintained in routine condition. It should not happen that if it is a media fill activity, so it is a big activity, so instead of our routine 2 times cleaning, we are doing 3 times cleaning, or we are doing some extra precautions or extra cautions which are not been executed in our routine activities. So we need to exercise the same level of cautions and precautions, and cleaning activity as well as environmental conditions shall be also be maintained as per the same standards as we are intended to maintain at our commercial production, so that we should be able to get our exact results of the media fill simulations. Cleaning and sanitization activity shall be done as per SOP. Number of operators in the area shall be same or more. So if in our routine activities, if in our routine production, if 2 operators are going to remain all the time during commercial activities, so minimum 2 operators should remain inside the area. It can be more than 2, but it should not be less than 2, because more than 2 it will be a worst case. Then run size and media fill frequency. So minimum 3 runs shall be done for a media fill qualification. Media fill requalification on each line shall be done on 6 monthly basis. So every 6 months we need to do a qualification of each line. Minimum 5,000 units shall be filled for in each run, and if a batch size is less than 5,000 unit, then actual batch size need to be adopted for our media fill trials. As you can see that in your biotech products or in your oncology products, many times it is less than 5,000. So whatever is your actual batch size, in case if you are not having the commercial batch size of 5,000 vials, then whatever is your actual batch size, then that number of units you need to fill for your media fill activity. Then industry trend is to fill 5,000 to 10,000 vials in each run. So generally in industry it is a trend that they are using. They used to fill around 5,000 to 10,000 vials in each run. Then incubation of media fill units. So all the units shall be incubated in pre-qualified walk-in chambers. As we discussed earlier that this is a prerequisites that all your incubators or walk-in chamber shall be qualified. Set temperature of plus minus 2.5 degrees centigrade shall be maintained. Any temperature between 20 degree to 35 degrees centigrade can be selected for incubation depending on your justification. Total incubation period shall be of 14 days. First 7 days of incubation shall be done at 20 to 25 degrees centigrade, followed by next 7 days on 30 to 35 degrees centigrade. Then interpretation of media fill. So if you are filling less than 5,000 units, then no unit shall be contaminated. If one or more units are contaminated, then we need to do revalidation for the media fill activity. If 5 to 10,000 units are filled, then if one unit is contaminated, then investigation shall be done including consideration of media fill, and if 2 contaminated units are found, then revalidation with investigation shall be done. If we are filling more than 10,000 units, then if one unit is contaminated, then investigation shall be done, and if 2 units are contaminated, then revalidation shall be done along with investigation. Then interventions of media fill. So this is very important part of media fill, that what are the interventions we are going to keep in media fill. Generally interventions are known interruptions which we are going to keep in media fill, because generally these interruptions are happening in our routine commercial production lines. So our intent is that it should not happen that any of the interventions we are not simulating in our media fill activity, and it is giving some adverse impact on our commercial production. So that interventions need to be evaluated. That's why we are simulating all the interventions. So what can be the possible interventions? We will discuss some of them. Loading of containers and closure on line. As we know that in one time we are not able to load all the containers and closures on our filling machine. So multiple loading of containers and closure it will be there. So we need to identify how many times we need to load containers and closures, and accordingly it should be done. Machine stoppage. In between our machine stops for lunch times or tea breaks or something like that, then it need to be simulated as per our routine practices. Then change of operators. If we are having an SOP that our operator will change it after 4 hours or after 6 hours, or during lunch time second operator is taking over, or during shift change second operator is taking over, so that activity should be simulated in media fill. The lunch and tea breaks as per routine shall be taken. It should not happen that in our commercial production we are taking lunch and tea breaks, but in your media fill we are not simulating that. So it should also, lunch and tea breaks should be also taken during media fill activity. Then operator change we have already discussed. Shift change activity. So during shift change our operator is changing, as well as if some other activities which are mentioned during which we are doing in commercial production, so that should be executed as per SOP. Then gloves change. So generally people used to have a SOP that after certain period of interval we need to change our hand gloves. If there is certain practice like this, then the same shall be simulated during your media fill activities. Cleaning of broken containers. So whenever our line runs, there is a possibility that few glass containers or will break, and there will be intervention, and we need to stop the machine. Then we need to clean that broken containers, and then again our line will restart. So that thing need to be simulated, that if our container is breaking in between, then what are the activities we are doing as per our SOP, same shall be simulated during your media fill activity. Then routine area and machine cleaning. So whatever routine activities we are doing in our commercial batches, same activities we need to do for machine cleaning as well as area cleaning. Change of filling tanks. So whatever filling tanks are there, it might be possible that we are filling 5,000 or 10,000 vials. So one tank will be sufficient to fill 5,000 or 10,000 vials. We filled with one our filling tank, but in our routine batches it might be like the maximum batch size is 200 liters or 500 liters. So multiple tanks are utilized for filling of your complete batch. So whatever number of filling changes are there, filling tank changes are there, so that need to be simulated. If it is a 4 changes or 5 changes, then those changes need to be simulated during your media fill activity. Then in process sampling. So whenever our commercial production runs, so we used to take the periodic in process samples. So same in process samples need to be withdrawn and checked for whatever test it is applied for. Then routine surface and environmental monitoring. So routine surface and environmental monitoring, as we are doing as per our SOP, so same shall be followed here also. And if any additional monitoring is there, so as per our protocol, so that need to be also executed. Then repeat charging of stoppers. So your container is stopper charging, it cannot be done in one time. So whatever repeated charging frequency is there, so that need to be followed for media fill simulations also. Then simulation of power failure. So if it is UPS line is there, then this activity can be avoided. But if any of the part is there on your direct power and UPS line is not there, so this power failure simulation can also be simulated in the media fill activity. Then we will talk of execution of media fill. This we discussed about elements of media fill, then your interventions which we are going to take up in media fill. Now we will talk how actually the media fill activity will be executed. So media fill protocol shall be prepared, that we discussed earlier also, that whatever activity we need to perform, first we need to prepare the protocol. Then along with media fill protocol, media fill BMR shall be prepared for each run, because each run is taken as a lot or one batch. So media fill BMR shall be prepared. Then training of media fill activity shall be provided to all the concerned personnel. So whatever operators are there, staff persons are there, then your quality control or microbiology people are there, quality assurance persons are there, in process quality assurance persons are there, then your engineering persons are there who are going to support in this media fill activity. So we need to provide the training to all the personnel for media fill activity. Then growth promotion test of the media shall be done. So your media shall be pre-qualified, that you need to ensure that whatever media we are going to use, that is qualified for growth of organisms. Then materials including media, containers and closures shall be issued. So as we are doing the batch processing for the media fill, so whatever material issuance sequence is there, that need to be followed. Media preparation shall be done in the manufacturing tank in manufacturing area. So whatever area which is given for your manufacturing activity, where we are manufacturing the batches in routine, so same area shall be used for your media manufacturing. Then containers and closures shall be sterilized as per validated procedure. So whatever routine sterilization process which need to be or which intended to be used in your commercial production batches, same process shall be used for your media fill also. Machine parts shall be sterilized as per the routine cycles. Prepared media shall be filtered through routine sterilizing grade filters. So whatever sterilizing grade filters which are used for our routine commercial production, same filters shall be used for your media fill also. Then integrity test shall be done before and after filtration. So as it is a regular practice in aseptic processing that before filtration of batch we used to do the integrity testing of your filters, as well as after doing the complete filtration again we are doing the integrity test of filters, so in order to ensure that our filters are intact. So same shall be followed during your media fill activity also. Then filling machine shall be assembled as per your routine practices. So whatever practice you are utilizing for assembling of a filling machine, same shall be employed over here. Then media in containers shall be filled and sealed. So once all these activities are done and your line clearance is done, then your media fill activity shall be started as per BMR, that is batch manufacturing record. Then execution of media fill. Next stage is your interventions shall be included during media fill activity. Whatever interventions we have discussed in our earlier part of this video, that shall be included in the media fill activity one by one. Media filled vials shall be visually inspected. As we are doing in routine production or commercial production activities, the media fill vial shall be visually inspected. Then visual inspection of the units shall be done by trained inspectors. Then next is visually inspected units shall be incubated at 20 to 25 degrees centigrade for 7 days. So once the units are filled and checked, then the incubation starts. So first incubation is done for 7 days at 20 to 25 degrees centigrade, and after 7 days these units are again visually inspected and then incubated at 30 to 35 degrees centigrade for 7 days. After completion of incubation, then units shall be inspected for growth. So during inspection of media fill vial, we need to explain to the visual inspectors during training that how the growth containing vials will look, so that they should be able to identify the growth in the media. Then some of the things what we need to remember is, if our product is having routine terminal sterilization cycle, then it shall not be considered during your media fill activity, because if we will do terminal sterilization, then we will not be able to find the contamination if there is any during our aseptic processing. So this step shall be avoided if it is there in your commercial production cycle. Then according to observation of 3 runs of media, the report shall be prepared. So once the 3 runs shall be completed for each line, and it is summed up, all the activities are done, then the report shall be prepared. In case of any contaminated units, then investigation shall be done. So as we have seen in acceptance criteria or interpretation of results, that if any of the contaminated unit is there, then we need to do the investigation. So whatever contamination, reason of contamination is there, we need to find how this contamination is coming, and then this investigation will include identification of organism and other reasons also, and identification shall be done up to species level to identify the root cause of your contamination. Then in documentation, what should be there in documentation? So naturally media fill protocol will be there, training record shall be enclosed with media fill protocol, batch records of media fill which we will be executing during media fill, it should be there. Media fill report shall be prepared based on all these activities. Investigation, if it is required, if we are finding any failure, then investigation report shall also be enclosed along with media fill report. Then sterilization records, sterilization of containers, closures, machine parts, that all the things need to be included along with report. Growth promotion test record of media which we are going to use for media fill activity shall be enclosed. Then environmental monitoring records, it should be included. Along with this, we can also include your water monitoring trends of that particular few days. Then filter integrity report shall be included into this. And temperature monitoring records of incubator shall be included, and material issuance record need to be included as a part of documentation in this particular reports. So friends, this was a very brief discussion about media fill activity or aseptic process validation. I hope it will help you in your understanding how we need to do the media fill and how this media fill activity is documented and how it is executed. If you like the video and if you find it is useful, then share with others. And if you are not subscribed my channel till date, then please subscribe the channel and press bell icon so you can get the latest notification for the upcoming videos. Thanks for watching.